Welcome back. We are being joined again by Dr. Claire Wade, and Mia and I get to have the opportunity to expand and discuss some of the things you just brought up. So first off, during your talk, the first part of your talk, a cat did sleep through it. We have Twitter documentation. A ferret was watching, and so we're getting a lot of feedback from the audience, so thank you very much. So one of the first questions I just wanted to think, looking back, it was noted that you moved from quantitative genetics to genomics.
Yes.
And could you tell us a bit about the difference between those two fields?
The two fields are extremely different. So I did my PhD in sheep quantitative genetics, and my PhD was concerned with developing more economically efficient breeding programs for sheep, and I worked in particular with reducing the costs of undertaking selection programs for sheep. So minimising the testing costs that were undertaken in order to make genetic progress for sheep. My PhD supervisor is a wonderful man, Professor John James, who still comes into the University of Sydney on a Thursday morning and has morning tea with me. He was a very classical Edinburgh-trained quantitative geneticist. He worked with many of the great quantitative genetics people in the world. And after I graduated from my PhD, I went to work as a sheep geneticist with the Victorian Department of Agriculture, where I tried to convince sheep breeders that they should undertake objective measurement and testing for their breeding programs. And I worked there with a guy who's also very famous, whose name is Dr. Mike Goddard. And the geneticists in the audience may have heard of him because he's involved with a lot of the new moves towards genomic estimated breeding values and genomic selection. After that, I took an academic appointment at the University of Queensland, where I worked to teach veterinary students about genetics. And at the time, there was a molecular genetics person and a quantitative genetics person. I was a quantitative genetics person. I stayed there for several years until I wasn't achieving the research goals that I wanted to, because I was quite isolated from the major quantitative genetics groups in Australia. I was working alone. I was not particularly well connected with industry, and I was not very satisfied with the work that I was doing. It's a really funny story, but at the time, the Vice-Chancellor of our university, who's the head boss of the university, said that he saw that the future of science was in genomics and informatics. And I went, I can do that. I know about genetics, I know about computing, I can put the two together. And so at the time I was due for a sabbatical, and the human genome had just been published. And so I wrote to the Whitehead Institute, who actually were part of the Genome Research Centre that actually published the human genome, and asked could I come there to do a 6-month study leave with them. And they said, please, yes, come, we need people just like you. So I went there, and I went there and they asked me what I wanted to work on. Did I want to work on humans? Or at the time they had human projects and they had the mouse project. And I said, well, I like animals, so I think I'll take the mouse, thanks, all the same. And so we did some really important groundbreaking mouse work there that resulted in first author Nature paper for me, and also the mouse genome paper. And the director of the institute, who is Professor Eric Lander, asked me to stay. And really, he's not a person that you really want to say no to at that. He says, Claire, you have to stay. It's a good time for SNPs. So I went, oh, okay. So I got in lots of trouble from my home university. I actually had to pay back the 6-month salary that they paid me while I was in Boston.
Gosh.
And I had to pay back the before tax component, which is higher than the after tax component. But it was worth it. It was well worth it for me, and I got to work on some amazing projects. So that's where I changed into genomics. Actually, I should say also, I should give credit to another great molecular person, is Professor John Maddock from the University of Queensland, who actually introduced me to the field of genomics through the Pseudomonas aeruginosa genome project. So that's how I got to Boston.
Wow.
I lived in Boston for 7 years, actually.
That's a trip.
Yeah, it's like coming home a little bit for me.
Yeah. Should we, you go back?
Okay, all right. Well, I guess one of the things that I am interested in, and also we've had other people expressing interest about it too, is things like when we're looking at things like working dogs, and not just dogs working on farms herding livestock, but also working dogs in, say, government roles like police and military roles, and also service roles like guide and assistance dogs. The language that gets used by the practitioners on the ground, so words like drive, that can be used across those different working dog contexts, but have very different meanings in those contexts, and even within a context by different people. I guess just asking about how useful or difficult as a researcher are terms like drive, when we're talking about desirable traits, and they say, well, I want a dog with strong drive. How do you manage that or deal with that?
It's a very good question, and I think even as you say, even among individuals, the use of the wording is quite different. So one of what we were hoping to achieve was to develop a lexicon, if you like, of common language in the industry, so that people, when they're talking about cast or talking about drive, I probably broke the rule myself, sorry, because I haven't looked at it for a while, but that they're using they mean the same thing when they're saying that term. And so if you say back, does that mean go back, or does that mean get on the sheep's back, or so things like that are very, and as you say, drive, what is drive? Is it a dog that works fast? Is it a dog that's keen to work? Is it oftentimes they're like
Persistence in times of
is it
difficulty?
persistent?
Yeah.
Is it resilience? Is it? There's so many things that encompass that thing that most people actually in working dog context see as being highly desirable. You want a dog that's really keen to get out there and get on the job, and keen to persist at doing that job all day if necessary. You don't want a dog that goes out and says, I'm going back to bed now.
Yeah. Well, that actually leads into a question that we got from Isabella, who's in Brazil, and she asked over Twitter, she said, thank you for your lovely lecture. In your opinion, how far should we genetically enhance the drive of working dogs without harming them from an ethical standpoint? So without having them work excessively, how far should we go in selecting for something like drive?
Yeah, this is something that I've given a bit of thought to, because you'll see in the next part of my work, my talk, where I explore this a little bit more in the retrieving context. In retrieving,
We don't want to steal your thunder.
I don't want to go into that talk just yet, but from a welfare perspective, there are issues in my mind, because I get concerned if those traits are overbred, that it's very difficult then to place the excess animals to requirements in pet homes, because they don't make good pets. I mean, well, they do make good pets, but they need to go to a very specific kind of home where you have someone who can manage a dog with that energy level, because they usually have very high energy level, very high need for exercise, very high need for novelty and engagement in their daily activities. So you can't leave that kind of dog locked in a crate all day and have it be happy. So, and those dogs might be the ones that end up in shelters as well.
Being displaced.
Yeah. So yeah, I thought about it. In terms of how far do you go, I had a conversation about this actually just last weekend with a retrieving dog person, and I was talking about my concerns about having more dogs than homes for the and they said no, it's no problem at the moment. They all get snapped up, right? But there's a bit of turnover. Like, you have people in those highly competitive areas that only keep a dog so long as it's performing, and then they rehome it, right? So it's fine as long as they can get a good home for it, but it concerns me what happens.
Well, we know having worked in shelters myself, there's a lot of Kelpies and Kelpie crosses that are coming through the shelter systems, both in regional areas and city areas.
Yeah, and a girl who was working with me on the cerebellar abiotrophy project, city girl living in the city house, has a little white fluffy dog, and because she was working with Kelpies, she got a Kelpie puppy, and it was just not she's done wonderful things with her, but not really the correct choice for a city dog where the owner's going to be away from home all day. So you really want a dog that is in a situation that's conducive to its welfare, I think. I think it's just another one of those things about, probably the biggest challenge is the correct placement of pups in homes. That's the hardest thing to do, I think.
Getting back to the foundational work that you've been doing with these two approaches, so we've got the top-down approach and the bottom-up approach, and we're trying to get at these questions about genetics and working dog behaviors. Tell us a bit more about, how do you know which approach to use? Do you use them both? Should they match up in the findings? Like, how does the general public think about these approaches?
Well, I think the top-down approach is one that's been used traditionally, you know, in phenotypic selection of dogs, and it's very effective. Like I always say to my students that one thing to remember is selection is extremely powerful. It's extremely powerful when applied accurately, which is one thing that well, this is off topic, right?
No, it's on topic.
It concerns me a little bit about estimated breeding values for hip dysplasia, because in a sense I don't know that I really want them to be selecting that accurately for it, because I'm concerned about it becoming just another selection for extremes. You know, I don't want dogs with 0 score hips if those dogs can't move. You know, I had a dog that had 0 score hips and he had sacral problems. So it's like, are you just swapping one issue for a different one? You know, is there a reason? The girl that worked with us on hip dysplasia, EBV's Bethany Wilson, had decided that actually in the retriever breeds, the looseness of the hips was likely to allow those dogs to swim. You know, because they needed more mobility in their hips to be able to swim properly in the water. And so if you've got these sort of combating breeding objectives where you want a dog that can swim and you want a dog with healthy hips, but you don't want the clinical expression of hip dysplasia.
Right.
On that note, Linda's asked a question through Twitter's asking, are there many breed organisations that are promoting quantitative genetics in their breeding programs?
I think it's becoming increasingly popular. Certainly in the UK, Tom Lewis was generating estimated breeding values for hip dysplasia there. Gregoire Leroy in France has been doing it as well. Australia's a little bit behind the curve with our ANKC population, but certain of the working dog organisations are certainly interested. As you can see, the Kelpie people are interested in it.
Here in the States?
I'm in the States. I think there's some move towards it through OFA, but they're not using the stringently accepted version of the estimated breeding values at this point.
So there's not really an international oversight.
There is. So one of the for hip dysplasia, it's a slightly different topic, right? But for hip dysplasia, the main issue is that no two countries have the same scoring system.
Oh, great. Okay.
So the OFA scoring system has like excellent, very good, good, poor, whatever. In Australia, we have one that's more similar to the UK, which is called the Willis scoring system, where we score each of 9 characteristics on each side of the dog on a 1 to 6 point scale. And then you tot up the scores and you give the dog a number. Okay. So for example, the average score for a Nova Scotia duck-tolling retriever in Australia is a score of 6, which is like 3 per hip. You want it to be 3 per hip or something. But you don't actually see clinical hip dysplasia until you get to a score of like 50. And and there are sort of like rough correlations between them, but
there's also pen hip scoring.
Pen hip score. So one of my dogs had pen hip done, and there's a lot of marketing surrounding the use of pen hip among the veterinary world. There's a lot of because there's a bit of financial incentive there, I think. But most of them correspond reasonably well with one another. But at the end of the day, you have to remember that those are indirect measurements of the thing that you're interested in, which is the clinical expression of lameness. And so saying that a dog of a hip score is going to be clinically affected is not really accurate, right? You can predict that a certain proportion of them might be, but you don't know. And likewise, you might get dogs that have 0 hip scores that will still produce puppies that have a score that's higher, and even from the best breeding programs, and that's just the nature of quantitative variation where the animals tend to spread out around the parental values.
Well, thank you so much, Claire. We're going to take a very short break and we'll be coming back for the second part of your presentation.
Thank you.
Thanks
Thanks
Mary
everyone.
Julie.
We'll see you again in about 5 minutes.
See you very soon.
15.